Every time you scrape your knee, grow even just a fraction of an inch, or, just exist as a uniquely complex human being, you're actually relying on one of the most spectacular, microscopic dramas in the universe. So welcome to this explainer. Today we're going to unpack the hero's journey of a single cell. We are looking at cell reproduction, specifically the epic battle of mitosis versus meiosis. It's this incredible story of exact cloning, of wild genetic diversity, and really the fundamental machinery that drives both our daily healing and our long-term evolution.
Okay, let's dive into this roadmap. We'll start with the life of a cell, then move into interphase, how it prepares to divide. From there, we'll tackle mitosis, the clonal split, before jumping over to meiosis, the diversity engine. And finally, we'll tie the whole shebang together by looking at why cell division actually matters to us.
All right, part one, the life of a cell. Let's look closely at the cell cycle and its timeline. Now, you might be wondering how this all works. Cell reproduction isn't just some sudden snap where one piece becomes two. It requires a highly coordinated sequence of growth and division. And what's absolutely fascinating is that the timeline for this varies wildly depending on the organism. To give you a sense of biological scale, look at this number, 24. A typical eukaryotic cell cycle, like say a human cell, takes about 24 hours to complete. Now contrast that with a simple yeast cell. That little guy races through its entire life cycle in just 90 minutes. The speed of life is incredibly relative when you get down to the microscopic level.
And this brings up one of the biggest misconceptions we have about biology. When we think of cells, we tend to picture them constantly splitting and dividing, right? But in reality, a massive 95% of a cell's life is spent completely outside of the division phase, in a state called interphase. Only a tiny 5% sliver of its life is spent in the M phase, which is where the actual physical division happens.
Which perfectly sets up section 2, interphase and the daily grind of the cellular factory.
So if a cell spends 95% of its life in interphase, what on earth is it actually doing all day? Well, it's definitely not just resting. It is working overtime, basically running 3 distinct factory shifts. First up is the G1 phase, or first growth, where the cell physically grows larger and synthesizes RNA and proteins. Next is the S phase, or synthetic phase. Now this is critical. DNA replication takes place here. The actual amount of DNA in the cell doubles, but, and this is a vital distinction, so keep this in mind, the chromosome number does not increase. You're just getting 2 identical copies tied together. Finally, we hit the G2 phase, the second growth, which is basically the final prep and quality control check before the cell is ready to physically divide.
But, nature loves an exception. Not all cells are destined to just divide forever. Some cells, like your adult heart cells, actually exit that G1 phase entirely and enter something called the G0 phase. Think of it as an inactive quiescent stage. They are essentially retired from the division game. They remain metabolically active, doing their vital daily jobs, like keeping your heart beating, but they simply no longer proliferate.
Moving right along to section 3, mitosis, the clonal split. We're looking at the equational division of somatic cells.
Okay, so what happens to the cells that do leave interphase? Well, the goal here is simple but monumental. Take one parent cell and create 2 absolutely identical diploid clones. Mitosis unfolds in 4 really dramatic chronological steps. First is prophase, the longest step, where tangled chromatin condenses into beautifully distinct, visible chromosomes, and the nuclear envelope just disintegrates. Step 2 is metaphase, this brief moment of perfect order, where these chromosomes align perfectly at the equator of the cell. Then comes the tension of anaphase, which is the shortest phase. Spindle fibers literally contract and just snap those sister chromatids apart, dragging them to opposite poles of the cell. Finally, telophase. The chromosomes uncoil and new nuclear envelopes assemble around them, forming 2 brand new daughter nuclei.
But we're not quite done, because the cell itself still needs to physically separate into 2. This is a process called cytokinesis. And I absolutely love this comparison because it shows how beautifully different life forms solve the exact same problem. In animal cells, which are relatively squishy, a cleavage furrow appears in the plasma membrane and acts almost like a tightening drawstring, just pinching the cell in 2. Plant cells, however, have rigid cell walls. They can't pinch, so instead they build a new wall from the inside out. Vesicles gather at the equator and grow outward to fuse with the lateral walls, creating a cell plate that eventually separates the 2 new plant cells.
So mitosis is basically a masterpiece of exact replication. It retains the precise chromosome number every single time.
But that raises a massive biological puzzle. If mitosis only creates exact clones, how do sexually reproducing organisms ever evolve? I mean, where does our incredible genetic variety come from if we're just built out of a biological copy machine?
The answer to that puzzle is waiting for us in Section 4, Meiosis, the diversity engine. This is all about reducing chromosomes to create unique gametes. While mitosis is all about keeping things exactly the same, meiosis is about shuffling the deck. It occurs only in our germ cells to reduce the chromosome number by half. And what's really fascinating about its structure is that unlike mitosis, meiosis actually involves two distinct sequential cycles. They're creatively named meiosis 1 and meiosis 2. But crucially, it only has a single cycle of DNA replication at the very beginning. So we run through a first cycle of prophase, metaphase, anaphase, and telophase, and then almost immediately run through a second, slightly different cycle of those exact same 4 phases.
Now, the absolute heavy lifting of meiosis happens right at the start in prophase 1. It is so long and so complex that biologists actually break it down into 5 specific microscopic subphases. We start with leptotene, where chromosomes become visible, then zygotene, where homologous chromosomes, meaning the matching pairs from your mom and dad, actually find each other and pair up in a process called synapsis. Then comes pachytene, which is a big one we'll discuss in a second, followed by diplotene, where they start to separate but remain linked at these X-shaped structures called chiasmata. Finally, diakinesis prepares them to move to the equator.
Okay, so the absolute most crucial point is what happens right in the middle of that sequence, during the pachytene stage. This is the definition of crossing over or genetic recombination. While those maternal and paternal chromosomes are hugged tightly together, they literally swap pieces of their genetic material. This is the exact microscopic moment where genetic variation is born. This tiny swap of DNA is the literal driver of human evolution. It's exactly why you are a completely unique mix of your ancestors. No one else has your exact genetic shuffle.
After meiosis I finishes separating those homologous chromosome pairs, the cell takes a very brief intermission. We call this interkinesis. And I really want to emphasize one crucial detail here. DNA replication does not occur in this phase. The cell is preparing for round 2, but it is deliberately skipping that S-phase copying mechanism so it can successfully have its genetic material in the very next step.
And that leads us to the final result, the magic number 4. Following that brief intermission, the cell plunges into meiosis 2. Now, this second cycle closely resembles standard mitosis. It simply splits the centromeres holding the sister chromatids together. Because the DNA wasn't duplicated during interkinesis, the final math results in the formation of 4 distinct haploid daughter cells. Each one contains exactly half the normal number of chromosomes, making them completely ready for sexual reproduction.
All right, let's bring it all home with Section 5, Why Cell Division Matters. We're talking everything from healing a scraped knee all the way to human evolution. We spent a lot of time zoomed in under the microscope today, so let's zoom back out. How do these invisible mechanics translate to the real macroscopic world you and I experience every single day? Well, it all comes down to their ultimate purposes. Mitosis is basically your body's dedicated maintenance and growth crew. It maintains your diploid genome perfectly. When you scrape your knee, it's mitosis that rapidly clones new epidermal cells to repair your skin. Meiosis, on the other hand, is the visionary artist. It conserves the specific chromosome number of our species across generations while simultaneously using that brilliant crossing over mechanism to create genetic variation. Think about it. Without mitosis, you couldn't heal a paper cut. But without meiosis, biological evolution would literally grind to a dead halt.
Which leaves us with this final thought. Just think about the incredible diversity of life we see around us today. The vast array of eye colors, our different immunities, our unique physical talent. Every single unique trait on Earth started with a microscopic crossing over event during the pachytene stage of a single cell. As meiosis continues its invisible work in populations around the globe, what undiscovered genetic combinations, what incredible leaps in human evolution are just waiting to be shuffled into existence in our future? It's pretty wild to think about. Thanks for joining me on this explainer. Keep wondering, keep learning, and I'll catch you next time.